today's workshop is going to be about how to use the Aquilos 2, preparing Lamella for Cryo-FIB. Hopefully everything's going to work. Yeah, so we'll be using Aquilos 2 Cryo-FIB, which is the second generation of its kind. And apologize for the interruption. So it's a dedicated Cryo-FIB tool. It's a small dual beam. Okay. I'm gonna keep it. That's fine. So it's a small dual beam. We have an electron column, ion beam column, and we have a stage which is a cryo stage, cooled by the cold gas running out of the viewer. So it's a dedicated tool to prepare to work on the samples which shouldn't be exposed to elevated temperatures, right? And we need to make sure we keep the temperature of the cryo stage below what would be vitrification temperature, which is about 140 degrees Celsius. So we need to be lower than that. And the technology allows us to do that. And on the right-hand side, you can see the transfer system, which allows us to transfer the grids, which would have biological cells on them without allowing them to warm up. So from your storage viewer through the transfer system into the microscope, where you would be doing your lamella preparation. And when you're done, you can use the same cryo-transfer system to take your samples back out. The Aquilos 2 Cryo-FIB has a longer runtime, so we can do automated lamella preparation overnight these days. It has an easy lift, cryo lift out system, auto slice and view, AutoTEM cryo, dedicated software to prepare lamellae in the fully automatic mode. Yeah. And these days, the IFLM, which is the integrated fluorescent light microscope, is an option, can be purchased. So quickly, you can see there is a SEM FIB column, which is at 52 degrees, not seen here. It's behind the IFLM setup. So we have the gas injection system, which is platinum, metal organic based. We have an integrated spotter coater built in, and a quick loader for the quick transfer of the samples. If you have the IFLM, this would be the specifications you would be getting for the system. So it's a wide field optical microscope. We have we have a light source which allows us to provide 4 channels. We can run it in the fluorescence and reflection mode, and switching between the modes can be done fully automatically. Yeah, thank you so much. Yeah. Yeah, thanks. The objective on the system is 20x 0.7 numerical aperture, which provides you the field of view around 500 microns at the working distance of 1.3 millimeters. We use dedicated IFLM shuttles for the work, which has 35-degree pre-tilt on it. It's a CMOS camera. It's fully compatible with the Aquilos 2 system. So we're gonna watch a quick movie. It's nice to show movies, right? Because they explain better what's happening inside. So we are looking inside the chamber of the Aquilos Cryo-FIB. I'm gonna stop for a second. Oops, sorry. Yep. So this is a cryo stage. It's fully rotatable, 360 degrees. And this is an electron column, ion column. This is a dedicated platinum sputtering device. And that's our shuttle that carries the grids that we want to work on. First, we want to deposit a protective layer of platinum so we can work with something's not right. Yep. Let me see. Yeah, I guess. Participants, yeah. Okay. Okay. Good. Yep. So first we need to deposit a protective layer, which is a GIS in this case, platinum GIS. And then we're gonna rotate the stage so that we could use the ion beam, which is coming from the left side at a shallow angle with respect to the plane of the grid. And this angle can be varied. It's called the milling angle. And as the process is happening, we can see what's going on with the SEM. So that's the advantage of the dual beam. So in this case, it's a frozen hydrated cell. It's quite thick for doing tomography. So we have to thin it down. And the way we do it is we place these patterns, so-called patterns. You can see they are shown in yellow. And we're gonna be rastering the gallium ion beam across and removing the material, ablating the material, so that at the end we are left with a thin region of the cell. So we can watch. Yeah, at this point, the lamella is supposed to be about 200 nanometers or so, which is suitable for cryo-ET. And the green layer is the protective GIS layer that helps us to protect the surface and help with curtaining, which is the artifact we have during the sample preparation. And at this point, it's ready to, sometimes it's optional to deposit a thin layer of metallic platinum for conductivity because our samples are not conductive. And now we're going to use the same cryotransfer system to, that's a coated lamella, to retract the shuttle, which contains our grids for further analysis. We're going to use a sort of similar cryotransfer system and introduce this grid into the Krios and collect a tomographic tilt series. So we are tilting the beam, collecting projections at different tilts, and then knowing the angles, we can do a 3D reconstruction of the volume. Yeah, so that's the movie. So a quick basics about the dual beam. So we are working with two beams. One of them is electron beam. The other one is the focused ion beam coming at 52 degrees with respect to the SEM. And two beams coincide at the so-called beam coincidence point, right? So we use the gallium ion beam for milling material and the electron beam for imaging. So on the right, you can see that's how it would be done in the case of, let's say, preparing material science samples, right? Creating a cross section and looking at the cross section. It's very important to be at the so-called Eucentric height. So on the left, you can see that's our sample. And let's say we are looking at this feature on top of the sample. If the sample or the stage is at the right height, providing this feature to be at the Eucentric height, that would allow us to tilt the sample so that the feature stays at the same place, which is important, right? We don't want to tilt the sample, which we have to do quite often, and that feature would move away, which is the case on the right. So if the sample is not at the right height, tilting the sample, just the feature would go away, and that's not what we want for the sample preparation. And quick basics about SEM, right? So we have electron source. We have lenses inside the electron column, which allow us to control the beam, shape the beam the way we want. So it's a very finely focused electron probe that we can raster across the sample. And at every point where the beam hits the sample, we have signals being for secondary electrons, backscatter electrons, all kinds of signals. And if we have the right detector, in this case, the electron detector, we can form an image, right? So for every position on the sample, we will have a corresponding intensity modulation on the screen. So that's a quick basic of the SEM. So by scanning across the sample, we are synchronously getting an image in our monitor. And the focused ion beam works similarly, except instead of using negatively charged electrons, we're using positively charged ions. Ions are heavier, so we have to account for that. But the principle is the same. The focused ion beam would be rastered across the sample, and the signal that would be generated as a result of the interaction of the gallium ion beam with the sample could be used to form a particular image. So why is it not? Yep. So we can see here on the left we have a sample. The beam is scanning across, and at the same time the image is formed on the right. So here we can see the point where electron beam hits the sample is not a point. It's actually what's called interaction volume. That's the area where our beam would be interacting with our sample. And we have to account for that. As a result of the interaction of the primary electron beam with the sample, we can have different types of signals coming back: secondary electrons, backscattered electrons, X-rays, and so on. So depending on what we want to do or what kind of detector we have, we could utilize that particular signal. And we should say that the higher the accelerating voltage on the column, the larger this volume, interaction volume, and the higher the atomic number or the density of the sample, the smaller it would be. So this is a footnote. And in the case of interacting, in the case of interaction of the gallium ion, which is positively charged with the sample, it's a destructive technique, right? Instead of, in addition to, let's say, gallium ions going into the sample subsurface and having an implantation, so to say, or creating different kinds of damage. In addition to that, we will have sputtering that's going to be happening, right? So every time we scan the gallium ion beam across the surface, we'll be sputtering some of that. So we have to be mindful of that whenever we expose our sensitive biological materials to the gallium ion beam. And that's the workflow for the AutoTEM cryo. First, we find the region of interest, put patterns where the ion beam will be scanning across. As a result of that, this material within these patterns will be removed. And the first step in the preparation, we call it a rough milling. We use quite high currents. And in yellow here is what would be the lamella at the end. So if we keep our patterns further away from this yellow region, we can use quite high current, relatively high current. And as we get closer to that yellow region, we want to reduce the currents as we mill our lamella. So we do the rough milling, fine milling, polishing, and as a result, we have a final lamella on the order of 200 nanometers or so. So some terminology that we're gonna use, that we're gonna see in AutoTEM cryo software. So in yellow, like I mentioned, it's a lamella. It has a dimension in X and Y, and then we have the height, which goes into the plane of the screen. And we have pattern offset, how far the milling patterns are from the lamella. And they have the size also. There is this called width overlap. When we start the rough milling, we want to have our pattern slightly wider in X. And as we get closer to the lamella, right, reducing our currents, we can reduce the size of the patterns in X. And this is called overlap. At different steps, it has a different value. And we can control all that. And in addition, we could mill trenches. You can see on the left and the right. It's believed they help to reduce the stress that gets accumulated in the grids when they get plunge frozen. But some users claim it doesn't help as much. So it's an optional thing to do to create these stress relief cuts or trenches. Oh no. Yep.
I don't get a good feel for how long this takes.
It's done.
How long does it take to get to the final step?
So it depends. It depends on the material, depends on the currents we use, and the size of the lamella we want to mill. I've seen papers where people would say, I've milled 15, 20 lamellae overnight, but what's the size we're talking about, right? Because we can have, I personally used to mill like 15, up to 15 micron wide lamella, and it would take longer. But if the X dimension of the lamella is like 5, 7 microns instead, yeah, you can have a lot more. On the order,
how many lamella is typical per grid? Just Ballpark.
Yeah, it depends. So now we're about to go on the microscope, and we can see, and we can talk about this detail. Yeah, yeah. So that's going to be our workflow for now. So the general workflow is to load a grid, take an overview in Maps, the software that we use for correlation or for navigation, evaluate the grid, select potential sites for Lamella preparation, spotter some platinum that helps with charging deposit protective layer, platinum JS, helps with curtaining, and then optional is another spotter code. And then after that, we can open AutoTEM Cryo software and run it. So at this point, the presentation is finished. So now I'm going to remotely connect one of the Aquilos 2 microscopes here. And you can see it. That's the user interface XTUI, right? So we have what I've done so far to save time, because we have a limited amount of time, is I loaded the grid. It's already sitting in the shuttle. You can see this is the shuttle. It's a bottom right quarter or quadrant 4. This stage is tilted to 45 degrees, which means having the sample sitting in the shuttle with the 45-degree pre-tilt makes it normal to the electron beam, right? So that's where it is. We are currently in the cryo conditions. You can see temperatures. So another way to see the temperatures here. So the temperature of the cryo stage and shield and pressure are shown here. The first quadrant, top left, controls the SEM. So we can see by this, oh, I'm losing my mouse, by this schematic. So top right is where we control the ion beam. Bottom left is the navigation camera, where you can see our shuttle from a different perspective, right? We have 2 grids loaded, position 1, position 2, 45 degree with respect to the horizontal. We will be working with grid 1, and with the next group, I'll probably work with grid 2. What else here? So we have a dedicated tab, AutoTEM tab here, or Snowflake tab, where we have the controls over, for example, mapping, milling, or deposition positions. These are presets that we can quickly go to. And currently grid 1 is in the mapping position, which makes the grid to be normal to the electron beam. And we want to be in this position to take an overview. So now I'm going to go to this kind of space. Yeah. So we have an external monitor that we can see. So top left shows the overview of the grid normal to the electron beam. Once we move to the mapping position, we can open the maps software. I've already done it, and we can create a layer. It's like the structure of the maps is like you can create a layer and import light microscopy data and correlate with your EM data. So in this case, I'm gonna just quickly switch. Okay, yeah, so we can see it a little bit better. Yeah, so grid 1, I took a snapshot, which is a low resolution kind of overview of the grid, and I took a tile set at a high resolution so that we could zoom in into the grid and see what it looks like and evaluate the grid, right? Before we decide where we want to mill lamellae, right? We want to see if it's suitable for milling. Of course, where the grid squares are broken, we cannot mill anything. If we zoom in, some of the grid squares will have cracks, so that's not gonna probably work for us. And we are looking for cells which are located sort of in the center of the grid square. That would be the best for tomography, right? If the cell is sitting like on the grid bar, probably it's not properly devitrified. And yeah, we're gonna be just milling the grid bar, copper grid bar. And what I've done is I've chosen a few sites which seem to be okay. But the way you do it, you just move around and evaluate what would be good. If you think this cell is good, you just right-click and add lamella site, and it populates here. So I've done a bunch of them, so I'm just gonna delete. So once you've done it, we need to deposit platinum, metallic platinum for conductivity, right? So for that, we have to go to the XTUI quickly. And so currently we are under high vacuum conditions. You can see chamber pressure, 2.1 -6 millibar. To do sputtering, we have to click prepare for sputtering. What's going to happen is it's going to go to a low vac mode to about 10 pascals. Yeah, and these are the settings we're going to use for sputtering. So the system will come to this pressure. And once the system is ready, meaning that the argon that we're going to use for sputtering has flooded the chamber enough so that we have this pressure, we can proceed with the next step. So the stage right now is moving to a specific location for sputtering. And there is a shutter that comes in to protect the detectors, which are in-column detectors in this case. And this is the sputter head that came in. We select the setting that we want to use, number 3. So it's going to use 20 milliamp current, this pressure, and we're going to run it for 15 seconds. We just click run. And the stage comes closer into the head. And then we'll see a flash in there, which indicates plasma. Yeah, right there. There was a flash inside. So yeah, basically argon gas gets ionized, and we're using heavy argon ions to hit the platinum target and sputter platinum everywhere, including our sample in the stage. Once this is done, we just wait for the stage to move down, and then we can recover from sputtering simply just by clicking this button, recover from sputtering. And now the vacuum system will come back to a high vacuum state. And when the vacuum is right, we can turn the beam on or open the valves to be able to proceed. And it's great, the stage moves to the position it used to be before moving to the sputtering. Yeah. Optional step would be to take another tile set after the platinum sputtering. Some users do that, but we don't have to. But we can take a look at what the grid looks like right now, right? Quick peek at that. We're going to click beam on or wake up the system. Yep. So currently we're using 2 kilovolt electron beam energy, 30 pkm current. And you can see by the change in contrast that the deposition has happened. So the next step would be to deposit a thicker. So this step was to help us with charging. It's a thin layer of conductive platinum. The next step is to deposit a thicker layer of platinum GIS. So for that, we also have to go to this Cryo-TEM preparation tab or snowflake. And there is a activity called Cryo GIS deposition, right? We want to do it on grid 1 and leave grid 2 for the next group. So we decide how long we want to do the deposition for. Let's use the settings that currently we have and run it. So grid 1, click start. So the stage now moves to this GIS deposition location. The GIS needle gets inserted to a specific location. The valve in the GIS opens up, allowing the hot gas come into the chamber, and basically it's a all the cold surfaces will be covered with hot gas, and there is going to be a layer growing up. When you know your deposition rate, it can be measured. You can then decide how long you can do the deposition for. Every user or site probably has done it. Yeah. I think with the 50-second deposition time. Okay, so what's the rate on this system? I think 400 nanometers per minute. 400 nanometers per minute. But it's a range, right? You can be within. When this is done, moves back, the GIS needle gets retracted, and we can quickly take a look at our grid, right? So again, the next step would be to take another tile set. So let's do that. I'm going to zoom in quickly, check the focus. Use the reduced area, focus on some feature which has sharp. Yeah, it doesn't have to be perfect. Okay, now we're gonna switch to display one or both. Yep. So this tile set was before sputtering. I'm just gonna copy that, call it after Python.js. And run. So if we zoom into that view, we can see that the tile set that I'm currently using is has the following settings. So it's a 5x7 tile set. The horizontal field width is 500. The resolution 12 times. All of this can be adjusted as needed. But we want to have a relatively good tile set because the AutoTEM software will take it from maps and use it for automation later on. So, and you can see as it's doing it, it goes from one to another. Do you have any questions in the meantime while it's acquiring the rest of the tiles? Yeah, you can see as it's acquiring new tiles, we will not see my Lamella positions because it's a layered structure. I will have to drag this new tile set down, and then we'll be able to see these locations on the new tile set. Yeah. Yeah, and we can watch what's happening in the first quadrant. The other parameter that I haven't mentioned in evaluating your grid is the ice thickness. If you zoom into one of the grid squares, if you see these holes in the quantifoil grid, right, that tells you the ice thickness is pretty small. If you don't see these holes, yeah, it tells you it's a little bit thicker. I've seen sometimes grids can be quite thick, which can be a challenge. But the idea is to work on your sample preparation until you have consistency in preparing your grids. And once you have that, you know that every time you get more or less the same ice thickness, then you can go for automation, right? Because then you can create a template that I'm going to show you in a second and use it consistently because you have consistency in the sample preparation. Yeah. Yeah, you can see that this bluish Lamella sites are not visible on the new tile set. Once it's done, I'm gonna drag this layer down on top of the one before sputtering. We don't do stitching. It's a, yeah, we want to have precise positioning. The stitching looks great, but not helping us. Yeah, if you had light microscopy data, you could import it, overlay with the overview we have right now, and decide where you want to do a targeted milling or Lamella preparation. Yeah, so almost done, 30 seconds remaining. And we don't have to acquire 5x7. We can have 2x2, 3x3, 4x4, or as large tile set as we want, or as small. Okay. The last one is almost done. Yeah, there is a lot to cover. Okay, so I'm gonna drag this new tile set under or above this old one. Kind of hard to control. Okay, so now we can see all the Lamella sites are positioned on top of this new tile set. So the next step is to open AutoTEM software. It's as easy as that. We're gonna come over here and launch AutoTEM Cryo 2.2. The latest version of AutoTEM Cryo software is 2.4, which we currently don't have installed on this microscope. So I'm gonna switch to this monitor. So AutoTEM Cryo is talking to maps, and you can see that there is this project in maps online that we can use. Basically, we can just double click here. And what's gonna happen is that, hopefully it's gonna do it. Okay, so you can see all the Lamella sites from the maps get transferred here, including from grid 2. We're not gonna work on all of them. We can just pick one on grid 1 and work with it. And the order in which the AutoTEM will be working on this Lamella sites is from top to bottom. So now it will be going to Lamella 16 on grid 2. We don't want that. We want something from, so I'm gonna just drag it here. Okay. It's not grid 1, I guess. Let's see, what would be and the other thing you may notice that before putting Platinum JS, we could see biological contrast, right? Our cells were charging and it was easier to see them. But now after the grid has been covered, it's really hard to see the cells. So that's why it's important to, thank you. Thanks so much. It's important to collect the tile set before sputtering and after sputtering. But let's pick up maybe, yeah, that one. Okay, Lamella 2. Okay, so I'm gonna deselect all. There's some lag in response. Let's see. Okay. Deselect all. Okay. And Lamella 2. And this is a tile from maps. We have a list of templates that you can build for your specific cell type, something. The AutoTEM Cryo software comes with the default yeast, default software, so it's sort of suitable for milling yeast. You can clone it and modify as needed and save it as a new template, right? So we have a bunch of different templates that you can modify. So the next step, once you are here and you have your list of Lamella sites taken from maps, is to go step by step, right? So the workflow is this. We are in the site list. We see the list of Lamella sites on the left. The next step would be preparation, which has the activities of setting up this site to Eucentric height, and then finding a milling angle that we specified here. In this case, it's, yep. And then once this is done, and we normally could do it manually or automatically, then we can go to the milling and the thinning. And the milling and thinning will be done fully automatic. But right now, we're gonna do a preparation step semi-automatic, right? So since I mentioned the milling angle, I want to quickly go back to the XTUI so that you can see again in this schematic. This is a nice kind of cartoon showing us the milling angle, the stage tilt, and the holder pre-tilt, right? So the stage tilt is 45 degrees. So that the grid, this is the plane of the grid, normal to the electron beam. This is the milling angle, right? That the ion beam makes with respect to the plane of the grid. In this case, is 38 degrees. Yeah, so it's really nice to have this schematic. Normally, milling angles can be anywhere from 6 degrees and above, depending on where on the grid you're milling or preparing your lamella. So let's go back to display one. So we want to run preparation for this lamella site. We click run, and you can see all the lamella sites. We're going to work on this one, doing preparation in the guided mode. We have a fully automatic mode, but let's do a guided mode. Let's click prepare.
Give us some expertise as far as the milling angle, why would you choose 6 degrees over 30?
Yeah, so basically, if you want to have a larger lamella in terms of like the higher the milling angle, right? So the higher the angle at which the ion beam is coming to your grid, the shorter the final lamella will be. So not in X, but in terms of like height. Yeah. So if you want to maximize the surface area of your lamella to increase the real estate, to have more till series collected on, you would go for that, right? But it takes longer again, right, to mill through. Yeah. And the other thing is that if you have a lot of ice on the grid, milling at a shallow angle may lead you with what we call shelves on the bottom, which means you haven't removed that ice on the bottom coming to a grid at a shallow angle. What the trick sometimes can be done is that to start your lamella preparation going with a very high angle of attack, right? Cut it to area, remove some of the ice, then reduce the milling angle. Continue, and then this.
Where the smaller angle and the rough cutting is at a higher angle?
No. In the software, you just we don't change energy. We use 30 kV gallium ion. Yeah. But if I see, I was going to mention that if I see a lot of a thick ice layer on the grid, it's probably not usable. I would just go and pick up a new one because it's not worth spending a lot of time doing all this machining, using ion beam. So we are in the preparation step. It wants us to place this crosshair on top of our cell. So that would be here. Right. And click continue. And probably I'm gonna go back to the entire view so we can see what's happening. Again, it wants us to place the crosshair where our cell is. We want to mill. Can confirm. It does auto brightness and contrast. So now we are in the step which does the eccentricity, right? And you can see I have this auto functions selected on the right. Electron, auto contrast brightness, ion beam, auto contrast brightness, autofocus. You can have them deselected. In that case, whenever the software decides to do them, it's going to do it. Otherwise, it will skip. Yeah, so back to our cell, click continue. And what the software is doing, it's tilting the stage in increments of 10 degrees or 5 degrees, and we can define and compensating any shift that happens to the feature from the center of the field of view, right? Trying to keep it in the center and calculating what would be the eccentric height at the end, which means when your cell is at the eccentric height, tilting the stage doesn't cause shift in your feature, right? So that's right here. And you can see how challenging it can be to see your cell after JS coding. Yeah. Okay. And that's our cell. Continue. Almost done. Yep. And you may have noticed the difference in material science, right? Field. When we find the eccentricity, we start from the 0 degree stage tilt, we tilt the stage and adjust. Here we are starting from 45 degree stage tilt and going the other way. So that's the difference you may have noticed. Okay. And it guides us, right? It tells us place the graphics where the cell is. Okay. And we're still in the eccentricity procedure. So now we are in the guided mode. We are working together with the software. It can be done in the fully automatic mode, but we need to be mindful in terms of what milling angle we selected for that site. Because depending on the location on the grid, the software may have trouble accessing it. So we are using AutoGrid, right? So it's TM grid clipped in the AutoGrid. And the AutoGrid has a cutout for the ion beam to go through to be able to mill. And if your lamella site is located outside of that sort of area on the grid, the software may have issues with that. So, okay, so it's now centering the site in the FIB. Yeah, FIB. Continue. So now you can see the feed view. We are using two detectors, ice detector and ETD detector, to help with shadowing. Yep. So yeah, that's our cell. Let's say continue. Yep, we didn't move much. Yeah. So the Eucentric tilt procedure is done. You can see the green check mark. So the next one is the milling angle. It wants us to tilt the stage to a particular milling angle. And here we selected 9. So on the lower right corner, we can see what's happening. So it wants us to tilt to a particular milling angle. For that, I would come to this tilt calculation and say, hey, I want 10 degrees milling angle. Type the number. So the stage tilts. I'm gonna refresh the FIB image. If it's out of focus or yeah, so maybe this is a good point to show. Low MAC, you can see it. This is part of the auto grid. And if I go to, let's say, 6 degree milling angle, for that site on the TM grid, it will not be accessible for this milling angle. So, but if you are further out to that part of the auto grid, it's still accessible. So depending on where you are on the grid, you decide on the milling angle you want to use. So let's just use 10 degree milling angle. Maybe, yeah, zoom in. And of course, we want to keep the edge of the auto grid. So you can see this is the cutout in the auto grid I was talking about. So for the ion beam to go through, you don't work at the limit, right? When the ion beam may be touching or interacting with the auto grid, which may be disturbing your beam, and the milling process will be not as good, I guess. So our cell is that cell. Yep. At this point, it would be good to focus, stigmate as you wish, and then come over to AutoTEM, update the image. Taking an image in XTUI and displaying it in AutoTEM, it will remember the coordinates. So now it knows this is the Lamella site, and place the thing. Yep. So the next step is to position our milling patterns. Again, all these parameters can be defined in the template, the size, and everything. So that's our setup currently. So the yellow is the Lamella side. So Lamella, you can see that it has the target thickness 250. So now I'm going to go back to single view. Yeah, that's better. So the target thickness of the Lamella is 250 nanometers. You can just type the number as you wish, and you can adjust the size. Let's say if you have more ice on one of the sides that you are afraid of would be blocking your view during the tilt series acquisition, you can extend the patterns one way or the other and make sure that that goes away. So I'll just put it here, and let's say we're happy with that, and click continue, and we'll say preparation is finished. So the next step would be set up the fully automated run. So you just come to milling. So these are the parameters from the template we're using. So that's the size of the Lamella, 14 micron in X, 5 micron in depth. The correction factor, so the patterns that the software uses, they are for milling silicon, right? So if I put correction factor 1, then all the calculations will be done as if it was silicon. Biological material is softer in a way, so that's why we use this correction factor 0.5. But again, it can be adjusted. This is one thing. That's one way to control how long it's going to mill at a particular step. So you just increase this number, it's going to stay longer and keep milling. If you decrease the number, so this is something to play with when you're just starting to use it. And then electron definition images, we don't need that. So the software needs a reference image in the FIB view that it can go back to and do the image matching. So we're going to leave it on. Stress relief cuts, yeah, we can leave them on as they are here. You can see the size of them. The depth correction is a local parameter to control how long or how long it's gonna mill at this point. So 0.5 is a global parameter telling the software it's softer material, but 100% for milling stress relief cuts is just that. And this can be experimentally determined. So the milling current we're going to use is 0.5 nanoamp. It's going to take another reference definition image in the FIB after that. And then it's going to go to rough milling using all these parameters. And you can see in this particular template, the depth correction is 75%, meaning that we realize that there is no need to have it at 100% because the material that we want to remove has already gone by that point. And we're using 0.5 nanoamp milling current. So you can see we're using quite low currents compared to material science application. And here we could select rectangular or cleaning cross section and so on. We have the rough milling, medium milling. So we are reducing the currents as we bring the patterns closer to our lamella and so on. You can see finer and so on. In the thinning step, yeah, we can do the polishing using 30 picoamp current. And at the end, I think some people use 10 picoamp current. Again, you can disable any of the activities you want just by toggling this off. So it will not do the thinning and we can start the milling and see what's happening actually. So we think we're ready to go with this lamella side. We just click run. If you are not happy with some of the parameters, you can adjust them on the fly. Just say, yeah, it's going to click run. It's going to take, it's an estimation of how long it's going to take, 53 minutes for milling. Yeah, so, but again, adjusting the depth correction will change this time estimation. But as the software starts working on milling, it will reevaluate how long it's going to take. So, and we have several options like site-wise or stepwise milling approach. Site-wise approach means it's going to select lamella 2 site, do the rough milling, medium milling, fine milling, thinning, polishing. When it's done with it, it's going to move to the next lamella site and do the next, so work on the site. Back in the days when the amorphous contamination of lamellae in the chamber were an issue, which is no longer the case these days, some users prefer to do stepwise approach. Like you would do rough milling for all the lamella sites, then you go to do medium milling for all of them, then and so on and so on. So that at the very end, when you do the polishing on all of them, your lamellae are polished, thin and nice, and you can take your sample out of the chamber and not leaving, to be contaminated with this amorphous stuff in the chamber. Okay, so we'll do sidewise in this case, click mill, and let's see what's gonna happen. Yep, so in the lower right corner in AutoTEM, we can see it says defining reference, and it's done. Yep, when one of the activities is completed, it shows it as a green check mark. So the next step is to mill stress relief cuts. And we can see in the second quadrant, top right, which controls the ion beam. Yep, so these are stress relief cuts. And again, it's optional how far you want to keep the stress relief cuts from your lamella. The idea is to relieve the stress, but at the same time to provide enough path for cooling to be happening, right? We don't want to accidentally devitrify our site. But yeah, so it's done. Stress relief cuts are done. It's taking another reference definition in the FIB because it has new features. It has now these 2 trenches on both sides. Yep. And at this point, when you're milling the stress relief cuts, you just need to make sure the beam cuts through the whole ice thickness, right? And that would be experimentally, again, determine how much of your depth correction you want to use for that particular activity or current, because we can go to a higher current and use a lower depth correction, or lower current, higher depth correction. So it's kind of interplay experimentally determined.
Now you can experimentally determine the depth of the ice.
But not really. But again, it depends if you have consistency in your sample preparation, right? And
Okay.
if it's not consistent, then yeah, even if you sometimes I see grids which would have like a gradient, ice thickness gradient across. Yeah, it's kind of tricky. It may cut through at this location, but it may not cut through the other location and do the job relieving the stresses. So yeah. And I've seen users have different templates for different cells. So one of them is yeast. The other one would be mammalian cells, flat, featureless cells. You can have bacteria cells. All kinds of. Yeah, you can see in the top right corner the bubbling of our layer. So it's a condensation that had happened, right? All the hot gas condensed on the cold surfaces, which is our sample also in this case. And now, and it's not cured, right? GIS in a way.
So
yeah.
Is it true? So let's pretend we're doing fluorescent detection of cells.
Yeah,
so that you're going to need
that's a great question. I'm going to go right to it. So now the next step, it would be just milling through, moving to the medium milling and so on, right? So now let's remote into a different
although that wasn't good my question, but go on.
Oh, okay. So okay, yeah, sorry about that. Yeah, I was going to show you this anyway. So that's on the other microscope, which is equipped with the IFLM. And we have John Mitchells and Alex Rigord attending the symposium. These are great resources to ask technical questions and other questions. But you can see here, it's a maximum intensity projection in both cases. And Jay, she was doing a targeted lamella preparation in this case. So what's in green is, I believe, mitochondria. Blue would be the nucleus of the cell, and the magenta beads for correlation. And the beads have different sizes, and I think some of them are inside the cell, some of them are outside. Yeah. Sorry, what was your question? I went.
Well, it was related to you saying that you really don't, that the surface, the ice layer is unpredictable, that your sample is somewhat unpredictable. And in my mind, I'm trying to come up with some way of automating some of this so that at least to the rough cut level, and then you could come back maybe give it fluorescent detection with the polishing and finishing up a couple of lamella that you're actually going to collect them.
Yeah.
Is there any way to do that, or is that just nonsensical? Is it going to be all manual for now?
Yeah, so the common way of preparing these grids is to use a VitraBlot, let's say from ThermoFisher, right? VitraBlot, or from a different company, where you plunge freeze your grid. But before you plunge freeze, you deposit your sample and you blot for a certain amount of time, or applying a certain amount of force, right, to the blotting paper. And it takes a little bit of back and forth to figure out your parameters.
Yeah, I feel like it's we'll go on.
Yeah. I agree. And it's an interesting question. But once you, let's say, have worked on your sample prep on that side quite a bit, in my experience, when we used to work on bacteria cells, they have a certain size, and you can have a quantifoil with different hole size, right? 2 micron or different. One extreme would be to have just glaciers on the grid, which means, it didn't, the liquid didn't go through the holes. It wasn't blotted properly. The other extreme, the grid would be totally dry, like you can see all the holes. And to get to this gold middle where you have a nice ice thickness with the proper cell density in the ice, so we could actually have some of them in your lamella, right? Final lamella. That's takes some time, I've seen. Yeah. So this is just one way of preparing samples, right? We can have a different shuttle to do work on HPF samples, which would be a lot thicker.
But I think that the answer to the question I was asking
Yeah.
is that we're really meant to the point that we can automate this.
Yeah, I agree. But once you have a consistent grid coming into your microscope, you can automate. the milling steps. Yeah, there is a lot of work, I guess, still on the sample prep, like everywhere else, like in material science, life sciences. Yeah, so I guess, yeah, I don't know how much time we have.
Yeah, I think we're actually just about finished.
But
We have like two minutes if there's any last thoughts or questions.
yeah, so you can see now it's still doing the rough milling, right? And time estimated is 46 minutes remaining. But what's important is to make sure at every step during the milling, all the material within the pattern is done. If anything is remaining, you have to deal with it later on when your lamella is already thin, which you don't want, right? Because you may have some backscattering, all kinds of damage happen to your ions can backscatter, right, and damage your lamella. We just want to make sure, yeah, it's a clear cut, meaning it wouldn't interfere with tilt series acquisition in the TM, right? And that's so before I would use AutoTEM cryo, I would just manually mill, right, and see, okay, with these parameters at this step, has it cut through or not? If it's cut through, I know that this depth correction is too much, so I can reduce it at this step and keep milling. If it's not yet done, then I would probably increase it or something like that. Once you have a good idea about your, again, assuming the ice thickness is consistent, right? Then I would start using automation in a way. So this is not the latest version of AutoTEM. The latest is 2.4. It takes into account the correlative work. So you can, in 2.4, you can create fiducials for correlation. So AutoTEM and IFLM can work together and do some good job. Thank you guys for attending. Yeah, thank you everyone attended remotely. Hopefully you could hear.